So now, the next talk is going to be great because now that you understand what UBE3A does, you can understand why we want to replace it. And we're going to have a talk by our next speaker here. Hold on, by Dr. Emma James. She's from Encoded Therapeutics, and she's not here representing Encoded. She's actually here because I heard her speak about gene therapy to parents at another conference that I was speaking at, and she just broke it down in such a way that was so incredibly digestible that I felt it was really important that you all heard that, because as you all know, gene therapy is a reality for Angelman syndrome today, and we'll talk more about that later today, as well as a ton about that tomorrow. But I want Emma to be able to explain to you what it means and how it works. So Emma, if you wouldn't mind, please take it away. Make this very digestible.
Good morning, everyone. If any of you see me talking more than once, you'll see I always carry my bottle of water around because I get crazy dry mouth standing in front of you all. But anyway, I would firstly like to thank Alison and the meeting organizers for inviting me here to present today. My name is Emma James. I work for Encoded. Encoded is a gene therapy company developing gene therapies for neurological disorders. My disclosures, I work for Encoded. So, as you've already heard today, we've come a long way in medicine. We've come from being able to develop antibiotics to developing vaccines that can save lives. And now we're really entering a new phase of medicine with treatment modalities that can go beyond managing the symptoms to addressing the whole range of signs and symptoms of certain genetic diseases. And at its simplest, the idea of gene therapy is that you take a cell with a non-functioning gene, you add genetic material, and that leads to the cell functioning normally. So I'm going to use an analogy here. So just like factories rely on detailed manuals to produce products, cells rely on DNA. And the DNA contains the instructions, think of it like blueprints, for building the tools our bodies need to function. So here we have the factory, and the factory has manuals, and the manual contains individual blueprints for individual products, a light bulb, a telephone, a security camera. Our cells are the factories. Our genome, which is made of DNA, is the factory manual, and the individual genes that comprise our genome are the individual instructions for individual products, which in the case of people are proteins. So the instructions inside this blueprint are really complex. And so the factory has a process to simplify them. So the factory, here it's a light bulb factory, and it copies or transcribes the relevant sections into a clear, step-by-step set of instructions. Think of it like an assembly guide. And then the workers can take that assembly guide and turn that into the product. So in the factory, it's a light bulb. In the people, it's the protein. So our cells work in the same way. So first, they transcribe the information in the DNA of our genes, and then they turn that into a simpler form, the assembly guide. That's called messenger RNA. That messenger RNA is then this clear set of instructions that is then translated into a protein. So, each cell contains lots and lots of sets of instructions, but it only chooses the sets of instructions that it needs based off the information it's getting from the environment. So each factory is responsible for specific products. So here, our factory makes only light bulbs. It has all the blueprints for all the different products, but it switches off the blueprints for the telephone and the security camera, and it focuses on the blueprints for the light bulb. And our cells work in much the same way, despite having the range of of all the different genes in nearly all the cells of the body, they choose, based off the signals that they're getting, which genes they're going to switch on. So in this way, a brain cell will switch on the genes to enable it to produce, say, an ion channel or a neurotransmitter. And it will switch off the controls that would enable it to become a muscle cell, say myosin. Similarly, a muscle cell will switch on the genes to enable it to be a myocyte, and it will switch off the genes that enable it to produce ion channels and neurotransmitters. And so this is the way that, that cells essentially are able to produce differential gene expression based off what that cell needs to be and what it needs to produce. So how does a cell know and control which genes it needs to be producing? Well, each gene has special tags that mark clearly where the instructions begin. And these regulatory elements are essentially binding sites throughout the DNA that help regulate both where and when the genes are expressed. So firstly, you have something called a promoter, which is a site in the DNA that regulates where the cell initiates transcription of a gene. And then near the promoters, we have sites for things called transcription factors. And these transcription factors are DNA binding proteins that help the cell regulate when or how much of the gene is expressed. These transcription factors can be positive or negative, so they can upregulate or downregulate, and they are cell-specific, again, enabling a cell to control what kind of cell it wants to be. So the promoter and the transcription factors work together as a complex on-off button to regulate gene transcription in a very tight manner, and they can be very, very highly specific to a cell. So the regulation of the gene, or the gene activity, can happen both at the DNA level, so DNA being transcribed into messenger RNA, which is that assembly guide, or the regulation can happen at the translational level, which is where the messenger RNA is copied into a protein. So now we're going to talk a little bit about gene therapy. So think of gene therapy as a delivery vehicle that goes into the factory with a new set of instructions to replace or change the blueprints that the factory is using. So gene therapy can be used to manipulate the gene expression and control the activity of the protein. It can work at the transcriptional level, so it can affect the DNA, or it can work at the translational level by affecting the messenger RNA. But the outcome is that you influence the protein that is ultimately produced. Gene therapy is an umbrella term for multiple ways to manipulate how cells work. Typically, when people think about gene therapy, they're thinking about when DNA is delivered to the cell via a single administration therapy. This could be gene addition, it could be gene silencing, it could be gene editing, and I'll come back to that in a few slides' time. According to the American Society for Cell and Gene Therapy, which describes gene therapy as the use of genetic material, or the delivery of genetic material to prevent or treat disease, RNA therapy would also fall under this category. RNA therapy uses shorter sequences of RNA to adjust how proteins are made, but typically it requires repeated dosing. Examples are antisense oligonucleotides, microRNAs, small interfering RNAs, and vaccines. What's a little bit more complex in terms of how we need to think about it here is while these microRNAs here fall under RNA therapy, There are now methods in development where gene therapy is used to deliver the DNA that provides instructions for the body to make microRNAs and small interfering RNAs, et cetera. So it can be a little difficult to really understand. In terms of the difference, if you think about spinal muscular atrophy, there are drugs that have been approved. There's one called Zolgensma, which is an AAV9-based gene replacement therapy. And that's really what people are thinking about when they think about gene therapy. And that is administered once. And then there is something called Spinraza, which is a chronically administered antisense oligonucleotide. And typically speaking, that's not what people think about when they're talking about gene therapy. We also have a third class here of gene-modified cell therapies. And this is a little bit different in that cells are harvested, they're taken out of the body, they are modified outside of the body, and then they're returned into the body. And examples of this are things like the CAR-T immunotherapies that we hear about for cancers, as well as things like lentivirus-mediated gene therapies, such as Lenmeldi for metachromatic leukodystrophy, which is another approved product in the US. So the delivery truck that we use to put the gene therapy into humans is called a vector. And vectors can be viral and they can be non-viral. Non-viral vectors typically require repeated administrations, and there are things like lipid nanoparticles and different polymers. Viral vectors are typically delivered as a one-time administration. And they've evolved to be probably the forefront of gene therapy because scientists have determined how to remove the virus's own pathogenic DNA and replace it with the therapeutic DNA. We know that genes are naturally evolved to be good at getting into cells. So this has proven to be a very good way of delivering gene therapy to the cells of interest using the virus's natural capability to infect cells. So gene therapy can be delivered directly to a person in vivo or ex vivo. So ex vivo is what I just described, where the cells are taken out of the body, they are modified in a Petri dish or a test tube, and then those modified cells are put back into the person. In vivo gene therapy is where the gene therapy is developed and then put straight into the person. And the route that you put in the in vivo gene therapy kind of depends on the disease you're trying to treat. For CNS disorders, often you will want to use a local route of administration, so maybe directly into the spine, or maybe via an intracerebroventricular injection straight into the brain ventricles in the brain. But there are other novel capsids. Sorry, I'll come to capsids in a moment. There are novel methods that potentially enable intravenous administration in development too. There's the dry mouth coming. So this slide here is a little bit old. This data is from 2022. But I think, broadly speaking, it's still approximately correct still today. So the most common viral vector used is called an adeno-associated virus. And in 2022, about 65% of clinical trials were using an AAV virus. Followed by AAV was a lentivirus. And a lentivirus is typically administered through ex vivo approaches. After that is an adenovirus. And what I want to highlight here is adenovirus and adeno-associated virus are completely different viruses. They are unrelated, despite the name. Adenovirus is a much bigger virus, and so you can actually deliver much bigger genes. But it's also typically more immunogenic, which means it causes more of an immune response, which is why AAV has evolved to be more popular. And then the non-viral delivery methods, such as plasmids and nanoparticles, are gaining in popularity too. I would expect a higher proportion of these to be used now. A number of gene therapies have been approved for use by the FDA. I'm not going to go through this. 7 of them are AAV-based, but there are also lentiviral-based and adenoviral-based gene therapies too. So what makes AAV appropriate for use as a vector in gene therapy? Well, importantly, even the wild type, which means the natural AAV, is not known to cause disease in humans. And they don't really trigger much of an immune response, which means it's less likely to cause side effects. So what we do, as I mentioned, is we take the virus, we remove the original virus genes, and then we replace them with new genetic material, which is called the transgene. The viral shell is called a capsid, and there are different kinds of capsids that have a different tendency to go to different parts of the body. This capsid is called the serotype. The serotype AAV9 has been shown to cross the blood-brain barrier, and so it has become one of the more popular gene therapies used for CNS disorders, but there are others, and there are also novel capsids that are in development too. One of the downsides of AAV is that they can only carry very small genes. It's a tiny virus. That means that some diseases caused by larger genes might not be treatable with conventional gene replacement therapy, which is where you just give a replacement gene, which means that novel ways to modulate the disease may need to be developed. So this is the case for Angelman syndrome with the UBE3A gene, which can't fit inside the AAV capsid, which means we have to have new ways of addressing it. So the AAV is a tiny virus, and it's taken into the cell by a small bubble-like structure called an endosome. It travels through the cytoplasm to the nucleus of the cell, which is the control center of the cell, which is where all of our DNA normally lives. Now, different gene therapies approach this differently. You can have some gene therapies that will, or some viruses will insert the genetic material into the person's genome. The AAV delivers the genetic material, and that genetic material forms this small circular shape called an episome. And it lives outside of your own DNA, so it doesn't integrate into the person's DNA. There are pros and cons associated with both. In terms of the transgene, the elements that we use resemble our own endogenous DNA. So we have the promoter, and that can be natural or synthetic. It can be cell-specific, or it can be ubiquitous, so expressed everywhere. Then you have the therapeutic transgene, and then you have regulatory elements that enable you to modulate up and down where and when the gene is expressed. So historically, most gene therapies have been used to deliver the missing protein that causes the disease. So that's called gene replacement therapy. But we can do much more than just replace proteins. So these are all, again, fairly broad categories, but we have the ability now to add functional copies, regulate, or silence genes to prevent disease. And really, the approach we use depends on the outcomes that you need and the fundamental basis of the disease, which is why it's really important that we understand the underlying pathology and the neurobiological processes of a disease. So gene replacement is where we give a gene, and it essentially produces a new protein. Gene regulation is where we deliver transcription factors that adjust gene activity by using these regulatory elements. So this is the approach taken by Encoded for our program ETX101 for Dravet syndrome, which is a haploinsufficiency, meaning only one copy of the gene works. So we deliver transcription factors that bind to the functional copy and turn up that missing copy, that extra copy, to produce more protein. Gene silencing essentially allows us to stop the translation of toxic proteins that cause harm. So with things like microRNAs, small interfering RNAs, short hairpin RNAs, essentially we deliver these RNAs that then bind to the messenger RNA and stop that messenger RNA being read and turned into protein. So think of it back to the assembly guide, which the workers need to be able to read and produce the product. And these gene silencing technologies take a black pen and scribble all over the assembly guide so nobody can read it, so that there is no product produced. And then gene editing is a little bit different in that we use what we call molecular scissors to go and edit the person's DNA directly. So Michael talked this morning about CRISPR, and that would be a gene editing technology. So essentially, we have a range of different mechanisms that we can use based off the disease that let us target different conditions with customized designs. One of the important things about gene therapies is that once you give it, we hope that it lasts forever, and you can't take it away. That means development of gene therapies is complex, and it involves a lot of interdependent factors. So it's absolutely critical that we understand the disease biology so we can design tools that address the underlying cause of the disease. As we've already heard, animal models are really, really important, not only to test safety, but also so that we can actually determine potentially therapeutic doses for human trials. Again, because we don't think we can give gene therapy again yet, I'll come to that in a moment, and you can't take it away, you want all of the doses that you give in human trials to be potentially efficacious based off the work that you do in animal models. So that animal research is absolutely critical and it has to be done very, very thoughtfully before you start humans. We need a robust understanding of the natural history of the disease. We need to, again, understand what are the signs and the symptoms that we are trying to address. We need to be able to then understand what is meaningful to the families and what do we think that we can change over the time course of a clinical trial so that we can select the right endpoints for the clinical trial. So the work that the Angelman community has already been doing in this regard to track and understand the natural history is incredibly helpful for people coming in to develop drugs. Clinical trials for gene therapies are complex. They are long, and because we are trying to, again, address the range and signs and symptoms, it means that we're assessing a lot, and families participating are really taking a big step in participating. It's a huge ask. Typically speaking, the primary endpoint, so the first time point at which you assess is the drug working, is usually over the course of about a year. And then the FDA usually requires that we follow patients for 5 years and then maybe up to 15 years. So again, participating is a huge commitment for families. Regulatory processes are a little bit different to traditional small molecules. I won't go into that here. And if you think about the difference between gene therapy trials versus those for anti-seizure medications that are just looking at seizure reduction, and they're just over 3 or 4 months, it's really important to have patient involvement in selecting the meaningful endpoints and what an appropriate risk-benefit the patient community is willing to tolerate. So it's really important to get your feedback when we're designing these trials. And then given the single administration aspect of gene therapy, our reimbursement systems really aren't set up to allow that. Insurance doesn't really know how to do that. So we really do need to be working with payers to evolve the commercial paradigms so that we can make sure that patients are able to access these therapies once they have regulatory approval. That's a whole different topic, and I won't go into that here. Another thing just to be aware of is that we still don't really know the optimal time for treatment. In CNS disorders, there is some evidence that earlier treatment may lead to better outcomes. It's going to be different for different disorders, and a lot more research is needed. But if you look at the figure here, no, I can't do that. How do I go back? Okay. If you look at the figure here, there's a dotted line on the left, just coming up, just under the word cognitive. That shows essentially neurotypical development, so unaffected children in a disorder called Sanfilippo syndrome. There is a red line there, which shows the natural history of children who did not receive a treatment. And what this figure really shows is that there is a child who was treated at about 1 year of age, and after 2.5 years of treatment, that child is still on the neurotypical trajectory. Children treated around 2 years of age are tracking also in that neurotypical range. And then children who are treated older than that don't have the same outcomes. Now, we don't expect this to be binary. We still expect that older patients will still be able to derive benefit. We just don't know how long it's going to take, and we don't know how much. So when you're testing a new therapy, you need to be sure that the effect you are seeing or not seeing is as a result of the therapy. That means that often in these neurodevelopmental disorders, we have to start young, because the youngest patients are the ones that we anticipate should be able to have the fastest responses over the time course of a clinical trial. And then once we've demonstrated that the drug works in those patients, then we can go into older patients to study that. It turns the traditional drug development paradigm on its head, because normally you start in adults and you work younger. But for neurodevelopmental disorders, where you're, again, trying to address the whole range of signs and symptoms of the disease, you need to start younger and then move older. But a lot more work is needed to be done on this to really understand what this means for each individual disorder. So when delivering anything to the immune system reacts. And this is the same for gene therapy. We want to be sure that, that we're controlling any immune response to that foreign material that's being introduced into the body. So there are sort of 3 different buckets that we think about. Because these viruses naturally occur, some people may have been exposed to the wild-type virus in the years before gene therapy. So those patients might have pre-existing immunity, which, if you then administer the gene therapy, the immune system may essentially mount a response and to stop it from delivering the therapy. There's also innate immunity. So immediately in the hours after gene therapy, the immune system, again, may see this virus and mount an immune response against it. And then you have adaptive immunity, which can happen in the days or weeks after gene therapy, where the immune system can develop stronger responses, like antibodies or T cells, that can attack either the gene therapy or even the treated cells. To prevent this, we test for antibodies before we give treatment, and sometimes patients who already have an immune response may be excluded, especially for the early stage trials, again, where we're trying to figure out, does the gene therapy work or not? We also use medicines called immunosuppressions to calm the immune response before and after. And then we monitor patients very carefully over time to manage any potential immune responses. So there are important considerations, many of which I've already touched upon here. Understanding the disease symptoms and progression is critical. And again, all the work you've done is really helping develop therapies because we understand Angelman syndrome. There are delivery challenges in hard-to-reach tissues. So again, if you want to go into the CNS, you may need invasive routes of administration. As one of the speakers already mentioned this morning, preclinical studies are vitally important, but animals are not humans, and they don't always predict what happens in humans. So we have to be very thoughtful and very careful before and as we start human studies. We are delivering billions of viral particles to patients. And so very high doses of this may cause side effects in the liver. So again, we need to be very careful. And these immune responses may limit future dosing or repeat administrations, which means we need to approach clinical trials a little differently because we can't do traditional dose-finding studies. And given everything I just said, long-term follow-up is needed. This is still a nascent field. I've been doing this for over a decade now, and we have a lot more experience than when I started, but it's still, we're still learning a lot. And so following people over time is going to be really important. But I don't want to sound overly negative. The data that is coming out is incredibly exciting. A lot of the approved gene therapies show a very favorable risk-benefit, and there is a lot of potential now for conditions that were once thought untreatable. On my last slide, given everything I've just said, development of gene therapies really does take a village. Sponsors and families and clinicians and regulators and payers all really need to be working very closely together to help advance these therapies forward. Thank you for your time and attention, and happy to take any questions. Thank you. Welcome.
Okay, I don't think we have time for questions. Thank you very much. So ultimately, I think what we learned today is that we start with things in a dish, we move to things in animal testing, we understand what we're putting into these drugs in order to try to replace UBE3A and how we may want to get it there, whether that's through a virus, whether that's through a lipid particle, whether that's through some type of editing carrier, all different ways that we're going to talk about over the next couple days. But I think what's also important to realize is that I know a lot of people just heard that talk and are super excited, but also heard that in certain examples of certain gene therapies that are having a profound impact on patients, almost all of the gene therapies where there's data, which Emma showed, is in neurodegenerative disorders, showing that if you address a neurodegenerative disorder before there are symptoms, you can traject on a normal neurodevelopmental trajectory. In a neurodevelopmental disorder like Angelman that is not degenerative, where we are on that trajectory and where we intervene and where we're going to see the greatest benefit is completely unknown. What the animal models actually show, if we want to believe anything, is that it doesn't matter what age you actually intervene, and you'll see that over the course of the next 2 days. You can see profound benefit at different ages, similar whether it's adolescent, adult, or newborn, in some therapeutics where you can really distribute well to the brain. And so it's really important that we don't get deflated. If we're not 2 years old, we're not going to see benefit. That's not what she was saying. She was just showing an example in a degenerative disorder in which you can not even see a symptom if you treat early enough before there's degeneration of the brain. But I want to acknowledge that in Angelman, for the example we have over the next couple days, we don't have degeneration of the brain, but we want to address the symptoms as early as possible so our kids don't have to live a long period of time with the symptoms of Angelman syndrome, and that's our ultimate goal. So I just want to make that super clear.